So I am very happy to introduce you guys to Dr. Suchinta Arif. She is an assistant professor at the University of New Brunswick in Canada. Um, she completed her PhD at Dalhousie University, where she was awarded the Dalhousie Doctoral Thesis Award. Her research focuses on developing models using a causal inference approach to understand how climate change and human pressures are impacting marine ecosystems. And though today's talk is focused on her work, I wanted to highlight that Dr. Arif is very involved in making the classroom and research space a more diverse and equitable place, particularly working with youth, BIPOC youth. I recently participated in her incredible workshop in the conservation, ICCB, so the conservation biology. Oh my goodness, the International Congress of Conservation Biology. And honestly, from there, I was like, "Ooh, I really have to invite her to U of O." So, um, without further ado, I leave her to it.
Thank you so much, Nicole. I'm super excited to be here. So as my talk today is titled Towards Causal Ecology, and just broadly across different disciplines, so health science, econometrics, psychology, and even ecology, we're kind of undergoing a causal revolution where we had this deep history of causal methods, and now we're kind of reappreciating these things as well as bridging them with contemporary and emerging techniques that together can help us answer fundamental causal queries in the sciences. And so today I kind of wanted to give you guys some tool sets that you guys can use moving forward, because yes, I can talk about my research and I'm happy to do so. And my research is embedded within this talk, but really what I want you guys to get out of it is something practical. So then taking it to the excitement that you feel for your research systems and your study systems, how can you apply some of these widely applicable transdisciplinary causal approaches? So kind of diving in to the paradox that we sometimes find ourselves in, many of our research questions are inherently causal, right? So as an ecologist, we might be interested in the impact that climate change or pollution or like multiple anthropogenic stressors are having on a population, an ecosystem, or even in human nature relationships, right? So anytime you ask these kinds of questions, you're asking a causal question. And even when we're skimming through the literature, this causal query kind of comes up even in the titles, right? So here's a title that I grabbed from Nature, Ecology, and Evolution: Social Environmental Drivers Inform Strategic Management of Coral Reefs in the Anthropocene. So when you see words like that, so drivers, influence, impact, that's all saying, hey, we're interested in a causal question. But we're kind of under a predicament where we have all of these fundamental and emerging causal questions that we would like to answer, and we're often reliant on observational data for doing so, right? Because it would be very hard to try to isolate the impact of multiple human stressors on entire ecosystems in a perfectly designed lab experiment. And that's not necessarily a bad thing because increasingly we have increased capacity to monitor and consolidate information from observational data. But nonetheless, oftentimes we are taught that correlation does not imply causation, particularly when we are working with observational data sets. So we're kind of in an interesting space where the tools, the statistical tools that we're often using when it comes to answering causal queries from observational data are not fit for purpose, right? They're not really meant to give you causal answers. And so, of course, we will be stuck with this correlation does not imply causation limitation. So today I'm going to introduce you guys to some causal tools that you can use to kind of get away from this mindset. So actually, it turns out that there are lots of valid causal inference techniques we can use so that we can answer causal questions, especially when we're dealing with observational data sets. So we're kind of bypassing this correlation does not imply causation limitation. So the first tool and the large focus of this talk will be around the structural causal model, which sounds very fancy, but its application is actually relatively easy once you kind of do the deep dive into it. And so the person that came up with this model is Judea Pearl, and so I've highlighted two of his books here. The Book of Why is more of a gentle philosophical introduction, and then if you really want to scratch your head and get frustrated, try the causality book as well. So essentially, we're just going to go through the main steps of this framework embedded in an ecological question that we answered, but hopefully you can take that tool set and apply it to your systems going forward. So because we're dealing with a room of ecologists, I'm of course going to embed this into the ecological question. So here we had a unique situation where there were around 21 coral reefs that were being monitored long-term in Seychelles. And you can kind of see a picture of a healthy coral reef there in 1994. And then around the 2000s, there was this widespread climate-induced bleaching event. And then through time, it showed that about half of these coral reefs recovered. So you can see a picture from 2011 of a healthy coral reef site that is recovering and has gone towards like the recovery trajectory. But the other half regime shifted to macroalgae dominated states. And so you can see that pictured in the other picture here. And so what's fascinating here is that we had standardized data, both spatially and across time. And so it gives us a really unique scenario to try to answer, well, what's leading to some of them recovering and others regime shifting following a climate-induced bleaching event. So that's our causal query. So what factors are affecting regime shift versus recovery trajectory following a climate-induced bleaching event across Seychelles coral reefs? So that's what we're going to try to answer. And we kind of just understand that this is inherently a causal question, and we are inherently reliant on the observational data to try to answer this. So under the structural causal model framework, the first step is to create a causal diagram, which is called a directed acyclic graph, or DAG for short. And essentially what this is doing is it's holistically capturing a researcher's understanding of the hypothesized causal relationships that are relevant for the research question. So again, we were interested in the different factors affecting regime shift versus recovery trajectory. So I know that this blue box here just says regime shift, but if it's not regime shifting, it's recovering. So it's really representing the two options there. And then we had a few different variables that we thought were going to be predictors. So depth, marine protected area, initial herbivore biomass prior to the bleaching event, and so on and so forth. So the variables of interest, so the predictors, enter into our causal diagram. And then essentially we have these directed arrows, which is the acyclic part of a DAG, that is qualitatively saying, hey, I think, for example, depth might have a causal influence on nutrients. And in turn, nutrients might have a causal impact on regime shift, right? So they're directed and they're going from cause to effect. And so a DAG should be synthesized based on prior domain knowledge, right? So this could include the researcher's knowledge base, as well as like synthesis from other studies that are inclusive of other experiments, other observational studies, theory, even like lived experience of coastal community members that are living and interacting with these ecosystems. And not even, but like particularly that would be useful as well. So essentially, you're kind of just synthesizing the theory on how you think that study system is working. And so here we talked to coral reef experts that have been working on the system for decades, and we also consolidated information from the scientific literature, and we were all kind of in agreement that this kind of represents what we think is going on. And so what's interesting about, and quite powerful about this, is that you're not really limited in what you're saying about these directed arrows. So yes, we're saying that depth can affect nutrients, but we're not saying if it's a linear effect, if it's a non-linear effect, if there's an interaction term, for example. We're just saying we think depth might have an influence on nutrients. So we're not saying anything about the functional form or the strength, right? So we're not saying that it's a strong effect, a strong positive, a weak negative. And after our analysis, it might turn out that depth doesn't actually have an effect on regime shift trajectory, even though we've hypothesized initially that there might be. So that's important. So just because we put an arrow in doesn't necessarily mean that we are asserting that it exists. We're kind of just hypothesizing that it might. So when you're putting in these arrows, you would generally want to put in the ones that you have enough proof or support that there might be a causal link, but you don't have to necessarily specify it much more than that. You also have to include common cause variables. So yes, we've included the predictors that we're interested in, but to make a DAG complete, you then have to kind of step back and ask yourself, are there any variables that are affecting two or more variables already in the DAG? And so if there are, you would kind of add that in. So imagine that there is a variable Z that is affecting nutrients, branching coral, and depth. You would have to add that in, right? If you have a variable Y that's being affected by regime shift, you would not have to add that in. So that's kind of just how you make it complete. And you do that to kind of account for all of the potential biases that could lead to that correlation does not imply causation bias. So once you have your DAG, you can then apply this very cool graphical criterion known as the backdoor criterion, which after you apply it will tell you, okay, to answer my causal query, given this observational data set along with this hypothesized DAG, what do I need to control for and what do I not need to control for to essentially take away all of those statistical biases that would otherwise lead to that correlation does not imply causation statement. So let's kind of embed ourselves in a specific causal query that we had. We were interested in the effect of nutrients on regime shift. So given this DAG, I want to apply the backdoor criterion to tell me what do I need to control for to answer this question. And so to apply the backdoor criterion, you need to block all backdoor pathways, hence the name. And so a backdoor pathway is any sequences of nodes. So those are the things in rectangles, so the variables, and arrows, so your directed arrows, that is connecting your outcome variable with your predictor variable, with an arrow pointing into both the outcome and the predictor. So here, just to situate ourselves again, nutrients is our predictor variable of interest, regime shift versus recovery is our outcome, and then we kind of have this one backdoor pathway, right? So it's going from regime shift to depth to nutrients. And you can read this any which way. So I could have said it's going from nutrients to depth to regime shift. So we know this is a backdoor pathway. So if we don't control for it, if we don't block it, it's going to potentially lead to a lot of bias. And so how do we actually block a backdoor path? You have one of two options. One is to control for an arrow emitting node in that pathway. So our only option here would be depth because there's only one intermediate arrow emitting node. And then the other option is to not control for a collider variable in that pathway. And so a collider variable is a variable with two arrows pointing into it. So imagine that instead of depth pointing out, you have regime shift going to depth and nutrients going to depth. In that case, we would not need to control for depth. But here we just don't have that option. There are no collider variables in that pathway. So to block this backdoor path, it's telling us that we need to control for depth. If that did not register yet, that's okay. We're going to try out an actual example where you guys will answer it for me in a second. So here, in this very simplified case, we know that our predictor variable is nutrients, our response variable is going to be regime shift versus recovery, And based on this DAG and the application of the backdoor criterion, we know that the control variable is going to be depth. And so we're going to get into how to actually control for a variable and how to build our statistical models a little bit after. But just to kind of introduce it, there are several ways to control for a variable. So if you think about randomized control trials, they're essentially trying to control for all of these confounders that might exist, right? Because the experimenter is setting the treatment at specific levels. So it's kind of taking away the impact of other things leading to confounding bias. When it comes to observational data, there's also a few different options for controlling for things. A popular technique that ecologists use is to add it as a covariate in your model. So if you have a linear model, you would have your predictor variable still be nutrients, but then you would add depth as a covariate. So it would be like another predictor variable, but you are using that as a control because then the coefficient associated with nutrients is saying, here's how nutrients is related to regime shift while controlling for depth. So adding it as a covariate is one option. It's not the only option. Another option would be like matching methods or even like stratifying the data. So you can imagine if we're only looking at a subset of data where the depth is kind of exactly the same, you're essentially controlling for depth in that way as well. Okay, so this is where we kind of do it together. And Nicole, I'm going to need your help in translating any answers. So here we're going to, so we looked at we looked at how to apply the backdoor criterion to get the effect of nutrients on regime shift trajectory. Now we're going to try it with branching coral. So you can see there's a few different causal pathways here. So we are expecting that branching coral is going to have a direct effect on whether a coral reef site regime shifts or not, or the amount of branching coral, or it can also indirectly affect it through its impact on structural complexity. So those are the causal pathways. But now I want to identify all of the backdoor pathways so that I can block them, and then I can get my adjustment set that's going to tell me what to control for versus what not to control for. So can someone, a brave audience member, give me a backdoor pathway? So again, this would now be connecting, it's a sequence of arrows and nodes. It doesn't matter which direction the arrows are pointing to, as long as the arrows are pointing both into our outcome, regime shift, or our predictor, which is now branching coral. So can someone tell me what a backdoor pathway is here?
Someone in the audience said depth.
Yes, exactly. So it's going from regime shift to depth to branching coral, right? So that's a backdoor pathway. I think that's the one I have. And so what should we control for? How do we block this backdoor pathway? So our options are to either control for an intermediate arrow emitting node or to not control for a collider variable. And so in this case, our only option is to control for depth. Does anyone want to try to find a second backdoor pathway?
Yeah. Wave exposure.
Yes, that's another easy one. I think that's the next one I have as well. So it's going from regime shift to wave exposure to branching coral. And so how would we block this backdoor pathway? What are we controlling for?
We control for wave exposure.
Exactly, wave exposure, right? So far, we've identified two backdoor pathways, and it's told us that to block all of them, because we do have to block all of the backdoor pathways, we need to control for depth and wave exposure. So here's another one. This one's going from regime shift to initial macroalgae to wave exposure to branching coral. Here we have options, right? So we can either control for initial macroalgae or wave exposure. And we kind of want to be as redundant as possible. So because we're already having to control for wave exposure to block that last backdoor pathway, this is already kind of accounted for. So we don't need to be controlling for anything else. And then here's another one. Here's another backdoor pathway, this time going from regime shift to herbivore biomass to MPA to branching coral. Again, we have two different options here. We could either control for MPA or herbivore biomass. So now we have two different sets of options, right? So to block all of these backdoor pathways so far, we can either control for depth, wave exposure, or herbivore biomass, or depth, wave exposure, and MPA. And anytime you have different options of adjustment sets, I would recommend going with the one that would have the least amount of measurement error. So in this case, we kind of knew if it was an MPA or not, whereas like I'm not confident that the quantity we had for initial herbivore biomass was the exact like representation of what was going on across those reefs. So I would pick the MPA one. Here's another one. So in this case, we have three options. We can either control for MPA, herbivore biomass, or initial macroalgae. But because that's already kind of captured in our two adjustment sets, and again, we want to find those limited sets of adjustments, we kind of just leave it as is. And then here's all of them together. And actually, it's fun to do, and I recommend that you do apply the backdoor criterion to your systems by hand, but don't actually do it in practice just because the level of human error that's possible is really sky high. So just as an example, I gave this as an exam question to my students for a midterm, and no one got the answer. And in hindsight, I gave them a really complicated question where there was like 10 like backdoor pathways. So like there's gonna be something you miss. So you can just do it online. If you go to daggity.net, it's a really user-friendly way to kind of just draw out the DAG, and then you kind of just set your predictor and your outcome, and it will tell you automatically what those backdoor adjustment sets are. And sometimes you don't have backdoor adjustment sets, right? So if we were interested in the effect of depth on regime shift, can anyone find any backdoor pathways there? So the answer should be no, because you don't really have anything pointing into depth, right? And so here, if we're assuming that this DAG is correct, you would just run a model with depth as the predictor and regime shift trajectory as the response. You don't actually need to control for anything else. And sometimes it is just as simple as that. Okay, so we've built our DAG. We're pretty confident in it. We've applied the backdoor criterion for each of our questions of interest. So I know we just went through depth and nutrients, but we were also interested in the effect of MPA, initial herbivore biomass, and so on. So we applied the backdoor criterion for each one of those models, and we're going to run a separate model for each one of those questions with the adjustment set that is specific for each one of those questions. So the next step is to actually figure out what that statistical model should be. And so here's where DAGs become very useful, because they're non-parametric, which essentially means they haven't made any assumptions, again, about like the functional forms or even like the strengths of those relationships. So you are actually very free to pick a statistical model that best suits your data and your question. So for example, if we thought that everything was linear, you would do a linear model. If you thought that some things were linear, some things were nonlinear, and some things had interaction, and you were like pretty confident about which ones had which, you could specify a GAM, for example, right? In this case, we went with a Bayesian logistic regression because our outcome is like binary. It's either regime shifting or recovering, so logistic regression made sense. So we applied a separate causal model for each one of these predictor variables of interest. And then these are kind of the standardized effect sizes we got. So because we coded regime shift as 1 in our logistic regression, anything on the positive side of this is saying that technically, like the more you have of these states, the more likely it is to regime shift. And then anything on the negative side is saying the more you have of those, the more likely it is to recover. So I'll just highlight a few findings. So we found that depth and structural complexity, if you are in deeper reefs or you have more initial structural complexity, you're more likely to recover. And this intuitively makes sense because reef sites at deeper reefs tend to be buffered against different stressors such as pollution and fishing pressure. And structural complexity provides habitat for like various different herbivorous fish that can then eat away at the macroalgae. Interestingly, we also found that initial macroalgae, so before the climatic event, there were like small amounts of macroalgae across the reefs, some with relatively more than others. And that like difference actually had a pretty big impact. So those sites with relatively more macroalgae prior to the climate induced bleaching event were more likely to regime shift, likely because there was just more starting macroalgae that were then able to take off with these new conditions. And the other interesting fact about that is that that wasn't even on our radar, and it only kind of came up through the DAG creation process. So sometimes just by going through the process of really like theorizing what the study system looks like qualitatively with those directed arrows can give you more insight about the different types of questions that you might want to actually answer. And it ended up being a pretty cool finding. And so around the same time this paper came out, our paper also came out, and they also found a similar result of that impact of initial macroalgae, but they did it through like an experimental setup. So at this point, what I'm trying to say is that I personally like believe in triangulation. I don't necessarily think that randomized control trials are superior to observational analysis, observational causal analysis. I think every type of causal method and or design has their strengths and assumptions and weaknesses. One is not necessarily superior to the other. You just kind of have to be cognizant about what makes the most sense for your question and your data that you have. And of course, anytime you have different methods and designs all pointing to the same thing, that just kind of is facilitating that that is likely happening. Okay, so you guys have done it. We actually just went through the whole framework, more or less. So again, if you are interested in the math behind this, like there's due calculus that goes into the backdoor criterion, feel free to like read that causality book. But what I appreciate about this method is that you don't have to dive into the due calculus. You can kind of just apply your theory and be in the driver's seat and like transparently walk through the process yourself. So going back to our effect of branching coral on regime shift trajectory, so what we did with our adjustment set is that we're essentially getting rid of confounding bias, which essentially arises when you have a variable that is affecting both your predictor and your outcome. So an example of this is if you look at the correlation between ice cream sales and number of drownings, there's like a very high correlation. And so I might conclude that if I eat ice cream, I'm going to drown, or like my chances of drowning is going to increase. But what's happening is that more people in the summer on really hot days are both eating more ice cream and swimming more, and hence the chances of the numbers of drownings in a country are increasing as well. So in that case, temperature is acting as a confounder. And if you don't control for that, you would get this really irrational conclusion, right? And so that's kind of what we did. So the backdoor criterion is telling us how to remove confounding bias. But I also wanted to highlight that there are other biases at play, because at least when I was being trained, I learned about confounding bias. And so there's like a lot of fear around it. And so you try to control for a lot of things to try to get rid of it. But unless you're like being very careful, you can actually over control for things. If you control for the wrong variables, that can also lead to biases as well. So one example of this is over-control bias, which can occur if you control for an intermediate causal variable. So going back to our question on the effect of branching coral on regime shift, if I controlled for structural complexity in that model, so if I included structural complexity as a covariate, for example, it would actually take away this indirect effect. So I would be biasing my results by kind of blocking this pathway, which I don't want to do, right? It's possible I do want to do that. If I wanted to look at the direct effect of branching coral on regime shift, that could make sense. But generally, people are looking for total effects, right? So you want to be very cognizant of over-control bias. And then you can also have collider bias, which occurs when there's a variable that is being affected by both your predictor and your outcome. So in this case, we also had data on juvenile coral density. And so juvenile coral density is being affected by branching coral through structural complexity, as well as like the trajectory of if it's like regime shifting or recovering, right? Because through time, you would have more juvenile coral density post climate bleaching event if it is recovering. And so that would also lead to that correlation does not imply causation. And so in the past, a lot of us have been taught to kind of have these causal salad models where we're putting all of our predictors that we're interested in under one model, and we're not really systematically going through what the control variable should be or what it shouldn't be if there's like over control bias that's happening or other types of biases, right? And so if you end up putting all of these variables into one model, it's going to lead to, given that this DEG is correct, it's going to lead to a lot of different biases, right? So one that I can see here, well, actually, maybe I'll open this up to you guys. So let's say that I put this all in one logistic regression, where again, my response is regime shift versus recovery trajectory. And then I just ended up putting all of these predictors as covariates. What kind of biases are we going to see? I'll give you guys a second. Do we see any maybe over control bias? I mean, I just named a few with respect to branching coral, so you can't necessarily tell me those ones. So we will see like our effect for branching coral will be biased because we have over control happening because structural complexity is here, as well as collider bias because juvenile coral density would be included. But do we see any other types of biases that might be affecting any of the other variables? Does anyone see any over control bias happening? For
Someone suggests nutrient and depth.
Yes. So if you're interested in the effect of depth, like the total effect, by including nutrients, you're kind of removing this causal pathway from depth to nutrients to regime shift. And what's interesting, though, is that when you're looking at the effect of nutrients on regime shift, depth is a confounder. So you actually do have to have nutrients and depth in the same model for nutrients, but you should not have nutrients and depth for the effect of depth. And that's why we kind of create like a separate causal model. It kind of just allows you flexibility to figure out what the control variables are, and then you interpret the coefficient associated with your predictor variable of interest. It also allows you more flexibility in the statistical model as well, right? So you might do like a linear model for one, like a GAM for another one, or like causal machine learning for another one. So lots of options. And so here's where I played around with the data a little bit. So sticking with the effect of branching coral on regime shift trajectory, here is our effect size at the top for our causal model. And then I did, here are the results for when I had confounding bias. So I think I took out one of the confounders from the adjustment set from the backdoor criterion. I did one with over control bias, so I included structural complexity. I did one with collider bias, so I included that post climate induced bleaching event, juvenile coral density. And then I did the causal salad model where I kind of put everything under one model. And the important takeaway is that like the relationships are shifting, right? So the model choices that you make will kind of impact the results that you get out of it. So I'm not necessarily advocating that I have the perfect DAG. Like you're allowed to disagree with my DAG, and that's part of one of the benefits of this approach is that you're being very transparent about your process. But I am advocating that given all of the modeling choices that we make along the way, we should be kind of intentional about it to the best of our resources and knowledge. And what's also interesting is when I ran AIC, which is often employed to select like the best model, it told me to use the causal salad model. But AIC picks the best model based on predictive accuracy, right? So what is the best and simplest model that will get me like the highest predictive, like in sample predictive accuracy? So which model feeds the most information on whether something regime shifts or recovers? That's fine if that's what you're trying to do. If I'm trying to predict it, then I would use AIC to select the best model. But if I'm trying to interpret the coefficients for a causal understanding, that's not really what that tool is meant for. But oftentimes AIC is being used for those two things. And so here's a model specifically, here's a paper specifically dedicated to that, which essentially is saying that predictive models aren't for causal inference because the two have been conflated. And in some cases they can be, but a lot of the times you just have to be very cognizant about whether the tools that you're using is actually fit for purpose. Okay, so that was my rant on the structural causal model. I do think it's super useful, and I think that it is widely applicable. But I didn't want to just introduce you guys to that. I wanted to throw in a few more. I certainly won't have time to go in depth into each of these, but I just kind of wanted to highlight that there are lots and lots of different tools that you can add to your causal toolkit. And essentially that would be the aim because it would empower you to be on the driver's seat and answer those causal queries when you have them. So one of the tools is instrumental variables. And instrumental variables becomes very useful if you have bidirectionality between your predictor and your outcome and or an unmeasured confounder that you just can't, you don't have it measured, so you can't really control for it. So here is an example. So imagine that we're interested in the effect of deforestation on malaria, and we're also interested in the effect of malaria on deforestation rates. But one is kind of affecting the other. So the level of deforestation rate it affects malaria, like the prevalence of malaria and vice versa. And so what we can do in this scenario is we can find an instrument. So to look at the effect of deforestation on malaria, we can have an instrument like commodity prices that is incentivizing deforestation rates, for example. And this instrument essentially has to be causally affecting your predictor and only affecting the outcome through your predictor, right? So commodity prices kind of hypothetically in this situation is meeting that criteria because that's not necessarily going to affect malaria except through deforestation rates. And then similarly, if we're interested in the effect of malaria rates on deforestation, we could use temperature, right? So temperature could be affecting the prevalence of malaria, but it wouldn't necessarily in this system be affecting deforestation rates. And so here is some results from an illustration from this paper that's in revision by Sam Walmsley at Dalhousie University, where he created a simulated data set where he knew what the true effect size is. And so you can see that the true effect size is the ones with the dash in it. And then he did a naive regression as well as like a IV approach. And you can see that the IV approach is much closer to the true estimate because it's not being biased by that bidirectionality issue. So just to say, instrumental variables has been used across other fields, and there's very creative instruments that people have thought of. And it has been used like a handful of times in ecological studies, but I'm very interested to see it kind of take off in ecology, just because I'm excited to see what like cool, interesting instruments that people can come up with, because they get kind of fascinating across other fields. So another approach I will highlight is causal forests. So causal forests get at heterogeneous effects. So far we've been looking at average treatment effects, right? So what is like the average like effect of this predictor on this outcome? But a lot of the times in ecology, those effects might vary across conditions, right? And that's kind of what we mean by heterogeneous effects. And so causal forest is a type of causal machine learning approach that you can apply, which essentially what it does is it works with the predictor and treatment, assuming that like the confounders are accounted for, and then you also feed it other covariates that are representing the conditions under which you think the average treatment effect might be affected. And it's essentially creating all of these decision trees that is splitting the data based on a threshold of one of those covariates that are representing the conditions, which is then maximizing the difference in the treatment and control outcome across the subgroups. And so it's doing this until it gets to these final leaves, and you have like hundreds or thousands of these trees. And so each individual point or case falls under one of these leaves, for which you have a conditional average treatment effect. And because you have like hundreds of these trees, you can actually average the conditional average treatment effect for each one of your individuals. So at the end, you can have this very cool graph where you have conditional average treatment effects for each individual case. So here's just one example. Here we were looking at the effect of depth on this kelp species, Laminera digitata, on the coast of Atlantic Canada. And the conditions that we put into it were latitude, longitude, as well as the abundance of invasive bryozoan. And so if we plot the conditional average treatment effects for each individual across space, we can kind of see this general pattern where the conditional average treatment effect is generally positive in the northeast and generally negligible in the southwest. And what we hypothesize is happening is that the northeast has generally more favorable conditions, which can allow for the general positive effect of depth, whereas in the southwest, the kelp there are kind of more stressed generally. So here, the relative positive effect of depth might just be overturned by all of these more important stressors. And you can imagine if we kind of average this to just like one average treatment effect, we wouldn't necessarily get that nuanced understanding. So those were just like two little points of additional tools that you can apply, but that's just three that we were able to cover in the 45 minutes that we had, right? So we went through structural causal model, instrumental variables, and causal forests, but there are lots and lots of both quantitative and qualitative causal approaches that you can use that have been like developed across different fields, and it's also like emerging as time goes on. So a very exciting time to try to be adding these toolkits to your resources. So with that, I think I will stop blabbing. Thank you.
If anybody has any questions, I can just repeat them back. Rebecca. You might, after you've already collected all the data
And I'm wondering if there's any particular things like incorporate stuff or any actual missing data that you find out later.
So Rebecca is saying usually this is meant for a study design phase. What happens if post study design you already collected your data and you find out that there's something that's missing and that you need, what do you do with that missing data?
Yeah, that's actually a great question. So it should be during the study design phase, right? Because then you know how to maximize your resources. So if you only need like a few confounders, there's no reason to be collecting all of these other variables if it's not necessary for your question. A lot of the times we do end up applying it to data that's already been collected because oftentimes we might not be collecting the data, right? Especially if you're using like global data sets. So there's a few different things you can do. There's lots of different causal techniques that can help you get around unmeasured confounding, right? So that instrumental variables was one, right? So that is one. You could also do sensitivity analysis, right? So if you have one unmeasured confounder and you think the effect of that might be negligible, you can try out a scenario under that. Versus if you think that it would be kind of strong, you can try out like a simulation under that. So there's lots of different options, and hopefully one of them will be ideal given your data availability and your structure and your question. But at the end of the day, even if they're not, and even if they are, I would just say be transparent, right? And it's just much better to still show the DAG, show that this was a confounder, show that you couldn't measure it, maybe show that you tried some other approaches that can either account for it or do a sensitivity analysis around it, and then just embed that within like your conclusions, right? So that the next person, if they really thought that confounder was important, that they would go out and kind of do it when they're designing their study.
Yeah. How robust are effect sizes if your DAG is not completed or misspecified? Sorry.
Yeah, so like the larger theory on this is that sometimes ecologists are looking for the model to give them the answer, whereas like here, the answer is really reliant on your assumptions, right? As it always is. Like the data often cannot tell you what the answer is, right? Whether you're using a causal approach or not, there's lots of assumptions embedded in your model. This is just kind of making them explicit. So I don't have a definite answer for that. Like sometimes if your DAG is wrong, your answer still might be correct because that adjustment set might have been exactly the same had your DAG been correct. Sometimes if your DAG is wrong and you're missing like a very strong confounder, then you're going to get a bad result, right? Like a biased result. But the strength in that is that you're communicating that very explicitly so that the person reading your paper can be like, okay, that's fine. I'm following this. This is not a black box approach. I can see how they went from A to B to C, why they picked their statistical model. I think they're missing a very profound confounder. Therefore, I'm not going to really trust that result in the way that they are, right? So it's not one thing. There is also this additional step you can apply to refine your DAG before you get at the backdoor criterion called DAG data consistency, which essentially, assuming that your DAG is right and that your data is relatively like measurement error free, there's a bunch of conditional independencies and independencies that should hold in your observational data. So you could use that to kind of additionally support your DAG as well.
Any other questions? Yeah, Peter.
Wondering how often in practice in ecology one runs into problems with there being cycles in the graph.
How often in ecology do you?
I think I heard that one. So how
Oh,
often do you have like cycles like bidirectionality or like feedback loops, right? because yeah, one of the things is that it's directed a cyclic graph, right? So often is my answer. But my follow-up answer is that it doesn't necessarily mean that if there is like a feedback loop in your system at large, that there's a feedback loop in the causal question that you're answering. And also you can employ time steps to kind of get over that. So there was actually a feedback between herbivore biomass and regime shift trajectory, right? But we used initial herbivore biomass, so before the climate bleaching event. And so if you use different time steps, even if A and B are affecting one another through time, you can still put A at time 1 is affecting B at time 2, which is then affecting A at time 3, and then still meet the requirements for it to be directed and acyclic.
Any other questions? No? Yeah, I guess with that, we can give our thanks to Dr. Arif.
Thank you so much. If anyone's interested, feel free to shoot me an email. I'm always happy to nerd out about causal models.
Thank you.
Thanks, guys.